Hey everybody, and it's time for another bridge video. Sorry for the delay. Obviously things have been pretty hectic with obviously first round of exams having just happened with many of you and with the previous classes, so advising was a little bit busy. But back on track now, so quick bridge video. You guys have been getting into your discussion boards and generating a lot of conversation so far with regards to epigenetics. Which is kind of this big topic now and leading into our deep dive of research that we want to get involved with now. So just a quick backtrack. Week 3, we were learning about gene expression or protein synthesis. So that's going to be a term that's used in the research a lot and is interchangeable. But with that, we were learning about how the DNA, the instructions to life, actually get expressed. So the genes are made up of the DNA, the A's, C's, T's, and G's, and then they get turned on or expressed, and they become proteins. And it's the protein that, once being made, is what gives us our phenotype, right? You know, why my hair is black, why yours might be blonde, you know, why my eyes are brown and yours are blue, green, brown eyes, etc. And so that's why we learned about, that DNA gets transcribed into messenger RNA. Messenger RNA is then able to move out of the nucleus into the cytoplasm, where translation occurs, because that messenger RNA then hooks up with ribosomes, which they themselves, remember, are made up of ribosomal RNA, or rRNA. And then, of course, the ribosome reads that messenger RNA in a 5' to 3' direction. And then, of course, as it's reading the triplet codon, so every 3 letters of that messenger RNA, that is going to attract and bring the transfer RNA over, and with it bring the amino acid. So, for example, if we're reading the messenger RNA, the ribosome is going to pick up on AUG, which is a start codon, which is the transfer RNA then is going to match up the AUG and with it bring methionine, MET, that amino acid. And then you start building the amino acids when a chain of amino acids is also known as a polypeptide. So we needed to understand that science and understand those basic fundamentals there of what DNA is and how it gets turned on, how it gets expressed, and that whole mechanism. Because now, as we look at the research from here on out throughout the rest of this class, we're going to hear these terms a lot. So you want to be familiar with the terminology of when they say protein synthesis, expression, genes, DNA, triplets, codons, etc. And when they're talking about how they're able to manipulate things in the labs, you'll understand why they're doing it, because it's based off of what actually happens in the cell, or what they're trying to mimic that is happening in the cells. So again, now as we're moving into week 4, we're starting to see that connection now between genetics and diseases, as well as starting to deep dive, as I said, into the latest research and what continues to go on. So that's why we needed that basic understanding. So now we're starting to look more at epigenetics and what that is. And epigenetics is really looking at also how environmental influences can affect us and get passed on from generation to generation. So just like having a gene that you've inherited from your parents' DNA that may make you a carrier for a disease, or may make you more prone, or maybe you actually have that disease. But instead of that influence coming from your parents' DNA, we're talking about how the environment around you is affecting those genes that you probably already have, but may or may not be turned on yet. That type of thing. So that's why we're also learning to see how DNA gets regulated, and those typical methods are through DNA methylation, histone modification, and non-coding RNA as well. And these are the kind of key ways that scientists are also trying to figure out how, through either drugs or genetic manipulation, can go in and, turn on or off certain genes for people who may be prone to a disease to prevent that disease from happening, or who maybe already have that disease and how to slow down its progression. So that's why we're looking at those things as well. So understanding that DNA regulation is important from here on out. And like I said, we're starting to discuss, and I want to see what you guys' interpretation is of the data and of the research is. So that's why we're going to be doing a lot of discussion boards, starting with week 4, almost throughout the rest of the semester now at this point. Because again, we want to see what you guys are picking up, how you guys are influencing each other in this class, and for you to see that, how this work in genetics and genomics is going to be impacting healthcare, both today and tomorrow, and when you all become official nurses, and how it's going to impact your interactions and what you teach and how you treat your future patients. So that's what's coming up. So that's week 4's bridge. And I will post something very soon for this weekend, probably tomorrow. Tomorrow's Friday, with regards to what's to come for week 5, because we're getting ready for one of your big assignments, which I think you're going to enjoy. It is going to require a little extra effort, and of course we'll still have a discussion board. All right.